Fantastic, thank you. So I'd like to invite the speakers up, and we'll open the floor up to questions. And as they make their way up, I'll just reflect that one of the things that really struck me across the presentation were the degree of tension. So there's the tension of the single nutrient versus the dietary pattern. There's the tension of genes versus the environment. There's the tension between understanding things at the population level versus personalized medicine and evaluating things through traditional RCTs versus the N of one study design, which is highly compelling. There's the tension between looking at what I consider to be final health outcomes, length of life, and quality of life, versus trying to identify better biomarkers along the way. So I thought this was a really thoughtful presentation, and I'm left with about 10 million questions, as I suspect others are as well.
Well, I can be the first one of that 10 million. I have a question. This is Fernando Brito with Nestle Health Science. I have a question to Dr. McFarlane. Thank you for your presentation, very insightful. There was one particular slide that I was very interested in when you were proposing some criteria of how to establish the distinctiveness of those nutrient requirements. There was one specific point that I was missing on that, which is the patient perspective. Meaning that we're talking about patients here, they have the burden of the disease, and we also have to consider if it's practical, possible, or sustainable for them to change their diet to meet those requirements. We know that certain diseases, the patients it's not even possible to meet the RDAs like the healthy population. So I would like to ask your opinion about that.
Thanks for the question. I think that's a hard one, because I think from, Health Canada is a regulatory body, and so ideally what we would have would be good standards based on good evidence of what disease responds to what particular nutrient intake. But I think that might be more of a question for healthcare providers. It might be more of a question for industry in terms of how they might formulate some of these diets or supplements. And it might be a question for insurance providers and what's reimbursable. So what's accessible, what's feasible. These are questions in the public health domain. How they might influence some of the considerations. I could see it in terms of whether it's compatible with oral intake and reasonable changes to dietary patterns and things like that. That might be a judgment that would be taken into consideration when setting these values. But I think your question is important, but maybe a broader scope than just this.
Bruce Burnett, I wanted to ask the entire panel, with regard to conditions or diseases where there aren't good biomarkers, do you have any advice, such as irritable bowel syndrome or even osteoarthritis, where we know radiographic measures aren't always predictive of symptoms? So in these cases, we have dietary interventions that seem to work with bioactive components or nutrients, but we don't have any reliable biomarkers to guide us in a design for future studies. Do you have any comments on that?
I can start. I think that we don't have biomarkers, perhaps because we haven't looked hard enough, and that was my point, in that we do have wonderful new technologies that will allow us to look at many different things. Let me just mention irritable bowel syndrome, since you brought it up. So there is a dietary intervention for the treatment of inflammatory bowel disease. It's a little bit controversial, but it's a low FODMAP diet. So if you were to ask the question, well, what happens to the metabolome on the low FODMAP diet, and you match it to bacteriotaxa, because these are basically fermentable substrates, or you ask what happens to the metabolome of plasma or in the feces, et cetera, you might be able to come up with biomarkers that might actually predict somebody's response to low FODMAP diet. Now, I'm not necessarily advocating a low FODMAP diet, but it is something that is out there in the literature, and it is dietary based. It doesn't tell you necessarily about the pathogenesis of irritable bowel syndrome, but there has been a notion that it is perhaps associated with an inflammatory process, a loss of barrier function, for example. And I think that we maybe need to revisit that. We collect biopsy specimens and we run it through mass cytometry, or we do deeper profiling technologies, and maybe that would help us understand the biological nature of that particular disease. Irritable bowel syndrome is tough because it's very complex, multiple phenotypes, many different reasons why people have gas and bloating and other symptoms associated with irritable bowel syndrome.
Yeah, maybe just to pick up on that a little bit, I think to discover such biomarkers, you might need, as I alluded to, better designs to get at causal relationships between, say, things that are measured routinely on individuals and outcomes of relevance. And oftentimes, very large studies are more observational, just looking for correlates, and that can have its day and is very important. But if there's not a design element to sort of separate the wheat from the chaff, the stuff that might be secondary to the intervention you're using, rather than kind of anticipate the responses or something. So design elements are often missing in studies where large amounts of data collected on individuals, so we could maybe identify true biomarkers instead of, secondary kind of associates of outcomes and whatnot.
Can I just, as this is an open discussion, can I just add to that, because it's a very interesting notion. It actually spins off to something I was going to ask Amanda about the level of evidence. So as a wet bench researcher that's primarily NIH funded, I always thought about research as sort of bench to bedside, basically do an animal model, try to determine its relevance in human biology. But increasingly so, we are starting to do these association studies where we're looking at biomarkers or things that are actually associated using these omic technologies. Now, we may not understand why these things are actually associated in human biology, but for a wet bench researcher, that's enormously valuable because we can bring that association back to the wet bench and try to phenocopy it in a culture system or in an animal model. And my notion is that if we can actually phenocopy it, maybe that tells us a little bit more about cause and effect relationships, the biological process that actually made those two things associated in human biology. And the value of that is that we already know that association exists in human biology. We just don't understand the biological principles behind it. But that's the interaction with the wet bench scientist. But I guess my question to you, Amanda, is the level of evidence. So if you do a study and you saw an association, a very strong association with certain biomarkers and a certain outcome, you still need some type of cause and effect evidence, I would assume. If you brought that back into the wet bench laboratory and you provide very strong evidence of a compelling argument that there's a biological process in an animal model that this actually occurs, do you then have to go back and prove that in humans, or is that going to be enough of a level of evidence?
I would argue that you still have to validate it in human, because I think the animal models, as someone who does animal studies myself, that's my primary research focus, they are critical for looking into the mechanisms that are underlying the associations that we're seeing. And I think I view science as an iterative approach. You see these associations in population data. It allows you to ask some questions. You go back to your animal models. You look to see what the mechanisms could be, and then you can validate those in your human populations. But I think without validation in humans, that there's a level of uncertainty that would be put into your modeling, and I don't know that that would be acceptable.
Even if the association actually already exists in humans because you started with human biology, if you knew the mechanism in an animal model, that's still not enough. You have to go back and do an intervention to prove cause and effect in humans.
I think you would still ideally do it in a human study. Okay. Patrick, did you want to follow up on that?
You want to make sure, obviously, that there is a cause and effect relationship. What came to mind for me is relative to certainty and mechanism. is what the WHO did when they looked at prevention of neural tube defects with folic acid. We still don't know why folic acid prevents neural tube defects. There were early clinical observations that if you gave one level of exposure, you prevented 70% of those. And then that became standard of care. WHO had to deal with the issue where countries were asking them, well, how much folate should we put in the food supply? And so there's a case again where you have an exposure, a black box, and you have a clinical outcome. And you have no idea how you came to that clinical outcome, but it worked. But they used a big data approach where they used observational data, big data sets, and created what was a computed dose response curve. So it was completely computed from observational data, and then it was validated from very small clinical studies showing, in fact, that that computed dose response was accurate. But there are no biomarkers that relate the exposure to the disease. It's just, from a clinical trial. But using big data, they could use observational data to get the dose response and then optimize that exposure to the outcome.
I would argue that they had the causal effect in an intervention trial first. So it wasn't an observational association study. So I just want to clarify that.
But the observational data gave you the dose response that you couldn't get from the clinical trial.
So let me agree with both sides of that argument and say for most nutrient studies, we will need validation in human intervention studies. But I think we want to be careful about being too dogmatic. And I'll use the extreme example of tobacco. Look, we had public health recommendations in the 1960s. We never did a randomized controlled trial of cigarette smoking and lung cancer, okay? We didn't need it. The evidence was so strong. There may be situations where we need to be, bend the rules a little bit. And the other thing is, we're in an era where if you write a grant, so much as you have to have a mechanism in order to make a recommendation. And I would argue it is nice to have a mechanism. You don't always have to have it if you do really wonderfully well-designed and well-thought-out clinical trials to show an effect. So I think we want to be flexible in how we judge each situation.
I just feel compelled to add that as a general pediatrician, if you showed me a large, well-conducted, high-quality randomized trial, or if there's a series of well-done conducted trials that show that a therapy is effective, I actually have I don't care at all what the mechanism is.
Neither does the parent.
Oh, were you there first? I'm sorry.
Claudia Morse from Memory. This question is for Dr. Wu. So as a parent of a child who was diagnosed with inflammatory bowel disease for years when I was in California, I was told by 3 very well-respected pediatric GI doctors that diet doesn't matter. I finally had to move to Atlanta. But so how do we bridge this gap? Because there's all this great evidence, but then just trying to bring it to the clinic and to educate our practitioners, it just reminds me of the very well-respected PICU doctors who don't think we need to feed patients for a week when they're intubated.
I will tell you that the number one question we as gastroenterologists get in patients with inflammatory bowel disease is what to actually eat. And I think our point, sadly, is we don't know exactly what to tell our patients. Now, Dale gave a really nice presentation about this. It's early days, and there is a level of evidence, but it's not really high right now because most of these studies are not well controlled. They're not very rigorous. These are sort of anecdotal, some a little bit better than that. I think that the defined formula diets are probably the best evidence that we actually have, and that's why we chose that. Our notion is that if we work really hard on this, and I think that that gastroenterologists and scientists finally have accepted the fact that diet does have an impact. And because now there is a sort of a groundswell of interest, and with that, more funding becomes available, I do think that we will be able to move forward in the field. And my notion is in a couple of years, we'll at least be able to give people some level of evidence and some recommendations about diet in a way that is very difficult right now. I will tell you the only dietary recommendations that we, in a standard way, give to patients with Crohn's disease that may have intestinal strictures is you stay away from fibrous food products because it could clog up your stricture and you could become obstructed. Short of that, we just don't have very rigorous evidence that most clinicians would feel comfortable telling people.
Yeah, Dennis Beer, Houston. I started to come up here with about one or two questions, and now I've accumulated another couple from the discussion, but I have two comments and two questions. The two comments are first, with regard to things like cigarettes and tobacco and no randomized controlled trials, there you're talking about published relative risks of 30, 40, 50, and 60, where you can be pretty sure that the signal is far greater than the noise. In nutritional studies, the relative risks are almost invariably less than 2. Given the noise of food intake assessments, given the noise of composition of diets, all the unaccounted for variables in the environment, frankly, I think the great part of the time when we see changes in 20% of risk, I'm not sure we know that those signals are greater than noise. It's a vastly different circumstance than tobacco. The other comment is that since I started the Victor Herbert Memorial meeting yesterday, I was on the FDA folic acid food subcommittee of the Food Advisory Committee when we voted to add folic acid to the food supply. Victor was at that meeting, and when the 10 studies were evaluated with regard to the effects on neural tube defects, at the end of all that, we've voted for first what the dose response should be to make a assessment of the fortification, and there was no uniform agreement on that except for that there were no votes for 0. So the span of how people interpreted that was very broad. In addition, we addressed the question of where was the level of toxicity adding folic acid to the food supply? When did we have to be worried? And the opinions of the audience, including experts like Victor and others and the committee members, differed by a factor of 10. So that's with the same level of evidence. What you do with it was in no way, uniform. All right. Then I have two questions. The first is maybe the simpler one for Dr. Wu, which is, you made very clear why you did the studies your way, because you're concerned about compliance as outpatient studies, and we've learned that, many years ago when we had clinical research centers before the NIH tanked them. And you had people as outpatients who, because you, I think, had reasonable, reason to believe they would, conform to their lifestyle diets. But I wonder if, if there's in other studies in normal people where we look at what happens between those patterns in the GCRC and outpatients with an idea of trying to assess noncompliance, which is a huge issue in nutritional studies. That is, what is the specificity and sensitivity of measurements we make in a GCRC with these metabolomic differences and very big differences in vegetarian and omnivore diets, and then moving to outpatients, continuing this, that there may be something in that will contribute, I think, an immense amount to, nutritional studies. So that's just a comment. I don't know if you want to respond to that.
Yeah, I'll comment on that. So our notion is to run studies in human subjects. We try to control as many variables as we can so that we can reduce the noise level and we see some type of signal, so some type of biomarker. But then we can use those biomarkers and do them in outpatient studies in a much larger category. The problem is that we did the inverse. we may never find anything. So our notion is, again, I'm a mouse researcher. A lot of things that we study in mice, sadly, especially with nutrition and diet, are not reproducible in humans in terms of the microbiome and the things that we're most interested in. But if we do this in inpatient controlled feeding experiments, at least we know that human physiology would allow those associations to occur. The question is, will that effect size survive the variability in an outpatient setting? Well, at least we know what those biomarkers are that are driven by human physiology. Whether or not that exceeds the noise that you'd see out in the population, I don't know. I think a fascinating study would be to take this omnivore vegan thing or other types of biomarkers that you see in the inpatient setting, do it in an outpatient setting in a couple hundred individuals, match it with their food frequency questionnaires or dietary recalls, and ask the question, how does it stack up to our current metrics of what we consider to be the best way that we can currently understand what people are actually eating?
By the way, I agree with that, with everything except the food frequency questionnaires, which are virtually useless. And I'm trying to move from those to giving us an idea of what the noise level is, and whether or not we can, in fact, make conclusions about what people do when they're not, locked up in a clinical research center. And I think there's either answer to what you just talked about is a valuable one.
I was going to say, before you get to your second question, I was just going to go over here just to make, because we're going to run out of time. But then if we have time after this, you can go back again.
Okay. My name is Wendy Johnson, and I'm from Nestle. And I have a question for Dr. Skork. I hope I didn't butcher your name. I really tried. Skork. Okay. So first of all, I was really impressed with your presentation. I thought it was amazing because it clearly showed the connection between public health and clinical health, because you used as your anchor the results from epidemiological studies to then measure how far patients were from their own norm. The question that I have has to do with how do we build the power in those studies in a way that we can aggregate that data and come closer to the personalized nutrition categories over time? And how do you do that across centers so that we can incorporate the variability that we need to make more robust decisions?
Great question. There's a couple of efforts that are being pursued to kind of bring people together. For example, I'm a member, and I'm sure others on the panel might be members of a CTSA site. So, for example, NIH has doled out about $500 million in a program called the Clinical Translation Science Award Program to build centers throughout the United States that focus on translational research. And the idea is to have those nodes in the network interact to collect data, share it. We could debate on whether this program has had success, but it's one effort, at least, to bring people together to collect large amounts of data that could be mined in the ways that I described. Again, that's just one example. I also think, just to your point about the public health thing and maybe some of the questions about RCTs, there are times when RCTs are the worst thing to do. So I'll give you a good example. Carbamazepine is a drug used to treat seizures. If a person has a particular HLA profile, they could develop Stevens-Johnson syndrome. Nobody wants Stevens-Johnson syndrome. Those connections between the HLA profile and Stevens-Johnson syndrome arising were from kind of clinical records and other things that physicians had observed. The idea that somehow you're going to do a prospective randomized trial subjecting people with a particular HLA profile to carbamazepine merely to make your point is insane. So really what we have to do is weigh the evidence in favor of something, and on that basis decide, do we have enough to go one direction or another? For many conditions, we don't have that kind of data, so we're going to have to collect more. I also mentioned that there was this movement afoot to kind of do away with phase 3 and phase 4 trials. But that's promoted on the belief that there will be a replacement kind of system in place, and we don't have that. And the system that people believe should be in place is collecting records on patients meticulously, collecting information that could be predictive of their responses or a particular outcome, recording that data, publishing it so that it passes peer review, and on that basis use the trends that are identified to help make decisions about patients going forward. So this is all a big data problem, and I think in an era of big data, this is all doable. We're not there yet, but I do believe, I was chatting with people about this in the back of the room before, the next generation is going to do this, right? I mean, I consider myself kind of old school in many ways, but I see a swell of interest in large data, mining it, being objective about it, not cutting corners, and I think this is definitely in our future. There's no question.
So it's your question.
Right.
Quick.
Well, I've changed my question to a comment.
Okay, go make your comment, and then we'll have a break.
This is really, I guess, in response to Dr. Shork, because all study designs have their strengths and weaknesses, and we could spend hours just talking about that. My concern with the N of 1 studies is that we have sort of the iconic example of the post hoc ergo propter hoc, fallacy, which has been known for several hundred years. And I would support that kind of study design if one predetermines what the outcome assessment is going to be and how it's going to be done, because right now there's an immense bias, with investigator degrees of freedom and post hoc justification of what you want to justify because you didn't specify it at the beginning. This is something which it's almost impossible to find a negative nutrition study, because that's exactly what happens with the post hoc, manipulations. And with regard to the algorithms, that's an hypothesis, it's a model. You can't ever prove that that's the mechanism. All you can do is disprove it. So you can get many studies that are consistent with this algorithm, but don't necessarily tell you causality at all. Right?
No, I agree with that. I think one of the points I wanted to emphasize, and maybe in the interest of time I just didn't have the opportunity to do, was the older kind of legacy RCTs that were pursued rarely collected enough data on any one participant in those trials to say unequivocally whether they responded to the treatment or not. So if you have a huge trial and 10,000 people, 5,000 get the antihypertensive, 5,000 don't, 5,000 get the nutritional intervention, 5,000 don't, and you collect a single measure, which has typically been done, or a few measures, you might be able to see average differences, say, between the group that got the intervention and the group that not. But you couldn't say, again, unequivocally whether any one of the individuals in the study responded due to the intervention. In order to do that, you'd need to collect the right kind of data. So the N of 1 study, the aggregated N of 1 study, are really meant to bring out the response phenotype in ways that are more compelling than you could do in standard RCTs where you just have a few measures on people. And in an era of personalized medicine and nutrition, this is really what we need to do, is to identify the responders and non-responders. Doing that with one or two data points is not going to happen. However, contrasting interventions to see if they respond to one or another, so you can, again, unequivocally state that they responded, this is doable, I believe, with the data that's available. So I'm not trying to poo-poo population studies. I guess what I'm trying to do is argue that if we want to decide who is responding and non-responding, we need to collect the right data on those individuals, and those are going to be more sophisticated population-based RCTs.
All right, you're going to sneak in.
Bernadette Mary at Medical University of South Carolina. Just on your to follow up, many of the states in our country are collecting or amassing very large data sets across hospitals of individual data. I think this is terrific because it is building our big data set. But that big data set is only as good as the people that we can get to volunteer to provide their data. So I feel as scientists we need to work carefully so there's careful protection of this data, and there's sort of an understanding of the public of the value that this data can bring by enabling their data to be put into large data banks and then used in this way.
Yeah,
but it has to be, they have to feel confident that their own personalized information will not be jeopardized as well.
Sure.
So now I'm gonna cut things off and take before I get fired. So I'm going to call it into the session. I'm sure the speakers would be willing to sit at the table while everyone else goes and grabs some food. And by the power vested in me, I give you 15 minutes. So we'll start at 10 minutes after.